I think many of you are aware that the Department and the services sponsor a wide range of medical and scientific research, and today we have with us a report on one significant part of that research. Captain Stephen Hoffman is with us today. He is from the Naval Medical Research Center in Bethesda, Maryland. He led a team of researchers in successfully testing a DNA vaccine on healthy humans, and he's going to give a briefing today about the Navy's work in this regard and their achievement in medicine's fight against many deadly diseases. Captain Hoffman has a statement to open the briefing, and then he'll be happy to answer questions on this subject. Captain Hoffman.
Thank you, Captain, and appreciate your being here. What we're publishing on tomorrow is on a new technology for developing vaccines. As many of you, I believe, are aware, vaccines are the most efficient, most cost-effective method of preventing infectious diseases, and in the case of the military, that also translates to biological warfare threats. The traditional vaccines that we have used really are not much different than the vaccine that was developed by Jenner for smallpox in 1798, which was a cowpox virus. And in the last 100 years, we have continued to make developments in the area of vaccine development. However, all of the vaccines that we have received are comprised of the foreign material, and that could be a weakened virus, a killed virus, a weakened bacteria, the foreign material that we are asking the body to respond to with an immune response, so that when the individual is challenged by the infectious agent, that infectious agent will then be eliminated because the body remembers how to respond to it. But in all cases, individuals are being immunized with the foreign material. With this new technology, which is called DNA vaccines, what occurs is that the DNA that encodes, in other words, the building blocks for that foreign material, is injected or is administered, and it can be injected, as we did in our study, but it can be administered perhaps orally on the mucosa and by many routes. And then that DNA, which codes for the foreign material, actually gets into the recipient cells where the foreign material is produced. And that is fundamentally different than all of the other vaccines that any of us have received. So the question comes up, well, big deal. What's the advantage of this technology over the old technology? I would mention at the outset that there are many important infectious diseases, in fact, the most important in the world, such as malaria, tuberculosis, HIV, for which we don't have vaccines. And there are certainly many emerging infectious disease threats and potential biological warfare threats for which we don't have vaccines. And one of the reasons, or several of the reasons for that, are one, that some of these infectious agents are very complex, and the technology that we have had hasn't been capable of inducing or eliciting the kinds of protective immune responses that we want. And the second is that some of these infectious agents, such as the virus which causes AIDS, HIV, change themselves very rapidly, and that therefore we have to attack it with many different immune responses at the same time so as to stay ahead of it. And although there are many groups working on using the more traditional technologies to develop such vaccines, we think that DNA vaccines offer a unique advantage in this area for one major reason and multiple other reasons. The major reason is that DNA vaccines are really easy to make, so that any molecular biologist, and that is the scientist who is a genetic engineer, anywhere in the world with a reasonable laboratory can make a DNA vaccine, and can actually make this DNA vaccine relatively rapidly, certainly on the month time scale, whereas to make one of these the more traditional vaccine often takes years. That means that in the long run, this technology is probably going to be quite safe, quite inexpensive or relatively inexpensive, scratch safe there, and we'll get to that in a second. It also means that one could rapidly make a new vaccine, so that if there is an emerging threat or a new agent that we didn't know about, making it with DNA vaccine technology is really quite easy. It also means that if you need to combine different targets into one vaccine, you can also do that very easily. And in the case of the disease on which I work, which is malaria, which is the most important infectious disease threat to our operating forces, which is a very complex parasite that has 6,000 genes in it, we can begin to build a vaccine that duplicates the complexity of the parasite by making many different DNA vaccines and mixing them together. That's not something we can do with any of the other current technology. So that the first major advantage of potential advantages of DNA vaccines are that they are easy to make, and thus you can make them fast, and you can make a lot of them, and they should be less expensive to make. The second potential advantage is that DNA is quite stable. After all, there are reports of getting DNA from mummies, and we've seen Jurassic Park where dinosaurs were, the DNA was extracted and dinosaurs were made from it. And that means that it's quite likely that this DNA vaccines would be stable under field conditions. That's particularly important to the military, and it's of extreme importance to getting vaccines out for children in the developing world, where one of the things that limits our capacity to immunize them right now is the lack of a cold chain. We don't know that for sure because this is only the first human testing of DNA vaccines, but our thoughts are that they will be quite stable, and thus you won't need this cold chain. The next advantage is relative to the weakened viruses and bacteria that all of us have received as vaccines, such as the measles vaccine, polio vaccine, smallpox vaccine when it was available, the recombinant smallpox vaccines that are being developed now for use against diseases like AIDS. There is always the possibility that a weakened virus or bacteria will revert and become pathogenic and virulent. With DNA, there's no possibility of that occurring. Now, DNA has its own theoretical risks, but one of them is not becoming virulent. The last advantage has to do with the other side of the equation, and it's something that we're reporting on in this paper, and that is the potential for eliciting a type of immune response which has been difficult to elicit with more traditional approaches. It doesn't mean that we can't do it with the more traditional approaches, it just means it has been difficult. And that particular immune response is killer T lymphocytes, cytotoxic T cells, which attack viruses, parasites inside of cells. Most of the vaccines that we've received are designed to produce protective antibodies, and antibodies kill infectious agents outside of cells for the most part, whereas in many cases the parasites, viruses hide within cells, and the way to attack them is through direct interaction with killer T cells. And in mice studies, the DNA vaccines have seemed uniquely excellent in that regard, and one of the reasons for that is because immunologists know that the best way to elicit this type of response is for the foreign material to be produced inside of the host cells. That's the best, most efficient way for the host to make killer T cells. I told you that what we do with a DNA vaccine is administer the DNA, it goes into the recipient cells, the foreign material is produced there, and that is the most efficient way that we know of eliciting this killer T cell response. So that, in summary, the absolute clear advantage of DNA vaccine technology would be its simplicity of production. We think that stability is going to be a major factor. We think that safety, as compared to the live, weakened, or attenuated viruses and bacteria, is a major factor. And the hope is that it will also provide excellent immune responses. And what we did in this study was using malaria as a model, because that's what we work on and that's what's most important to our program, we constructed, in collaboration with Vical Incorporated, which is a San Diego biotech company, a malaria DNA vaccine. Our initial goal was to test it for protection. To do that, we needed a complex vaccine, which was 5 genes that were available to us. When we went jointly, that is the Navy and Vical, to the Food and Drug Administration, they said this is a brand new technology. First thing you have to show is that it's safe, well tolerated, and actually can elicit the immune response you're interested in. So they said do it with 1 gene. So we went and did this study with 1 gene in 20 volunteers, and what we found was that it was safe, well tolerated, and elicited excellent cytotoxic killer T lymphocyte responses. So what we have done is prove the principle that this technology, which was first discovered in the and tested in the early 1990s, the first actual paper on this showing immunogenicity was published in 1992, the next in 1993 on protection of mice, and since then there have been a host of papers in mice, rabbits, and monkeys studies showing that that DNA vaccines were immunogenic and protective, but this is the first demonstration in healthy humans that in fact we can make the jump from our laboratory animals to humans. And as such, this provides the precedent for greatly expanded studies in humans testing malaria in our case, but in many other anti-infectious disease vaccines.
Is it more important that it's a DNA vaccine or that it's a malaria vaccine?
For this paper, it's much more important that it's a DNA vaccine. This is not, we used malaria as a model. What we have made here will never go forward as a malaria vaccine because it's not complex enough.
So give us some sort of explanation of, you worked with the malaria vaccine, but what other applications do you see?
There are groups around the world working on almost every other infectious disease that you can think of, and within the military there are groups working on biological warfare threats. So that there have been studies done in HIV-infected individuals, and in fact there were 2 publications earlier this year in HIV-infected people. The interpretation of those is difficult because they're infected, they're immunosuppressed because of the infection with the virus, and they already make immune responses, so you can't tell with complete certainty that what you see is due to the infection or to the vaccine. But so there's work going on to develop a preventive AIDS vaccine, TB vaccine, herpes vaccine, Ebola vaccine, Lassa fever vaccine. Pretty much everything that you can think of is now being studied, has been studied at least in mice or monkeys, and now studies are starting to go forward in humans. But the concern has been, is it really going to work in humans?
This isn't the first DNA vaccine testing in humans, is it?
This is the first report of any DNA vaccine in healthy humans.
So the human testing that's been done before has been in ill people.
The only other reports, as far as we know, and in the world's literature, are in HIV-infected individuals, and those were also reported earlier this year.
And that's the one where you say because did you get any T-cell response? I guess it's not your study.
It's
so
not our study, but in the study, one conducted in Sweden and one conducted in the United States, in the Philadelphia area, the University of Pennsylvania, and a company that was then called Apollon. The study in Sweden reported these cytotoxic T-cell responses also. There was, but the concern, not the concern, there was great enthusiasm for that result. Amongst immunologists, there's always the concern that, in fact, that might have been due to the infection as opposed to the vaccine.
would it generated a T-cell response?
Yes, the infection itself generates T-cell responses. Yes.
Yeah, I want to clarify something. You said early on that one of the advantages of the DNA testing is it could work on a complex virus such as malaria. But then you said that the vaccine the DNA vaccine that you tested could not go forward as a malarial immunization because it is not complex. So could you clarify?
Sure. Yeah. Because we were essentially first, or among the first, when we went to the Food and Drug Administration and said we want to test the complex vaccine because that's what we think is needed for protection, they said you have to go test the simple vaccine first because this is a brand new technology, and the first thing we have to establish in humans is that you can give them DNA vaccines safely. So they said test one first and then you can move to the complex one. So that's what we've done. In the process, we have shown that you can induce these killer T-cell responses at a magnitude which is good, if not better, than seen for any other vaccines.
So what you're saying is the simple the simple model will not necessarily protect a soldier in the field.
Right.
But it will give you
It gives
the
us
foundation to go to the next step,
absolutely
which the complex vaccine will eventually do.
right.
Will you develop will you develop a complex vaccine then?
We
or
are
not?
manufacturing with Vical right now two complex vaccines, one that has 5 genes and one that has 15 genes. And we plan to start testing the 5-gene vaccine next summer and the 15-gene vaccine several months later. And
Malaria.
these are both for malaria. My entire focus in my life is on malaria.
Explain the test that you have to do in order to prove that you have developed the malaria vaccine.
Okay. So the question was, what are the tests that you need to do to prove that you have a malaria vaccine? So that what we would do is immunize individuals, do the exact same study that we've done now, which is show that test it for safety, which is first and foremost with this new technology, show that it induces these immune responses that we're after, and then in the case of malaria, we actually expose the volunteers to the bites of infected mosquitoes. And the controls, the ones who didn't get the vaccine, get malaria, all of them. Because we do it, we have a lot of experience with that, and we do it in a very controlled setting, it's quite safe. The individuals become ill with a flu-like illness. We diagnose them very rapidly and treat them. But it allows us to determine whether or not the vaccine is actually protective. Eventually, we would have to then, having demonstrated that it was protective in this type of setting, we would then take it to a place where there's natural exposure to malaria and immunize individuals and determine whether or not it protects those individuals.
Will the mosquitoes used in the experiment be infected only with the level of malaria that you immunize these people on the 5 and 15 gene?
No, they have the full complement. They have everything, and the hypothesis is that by making an immune response against those 5 genes that you will totally knock out the parasite. And we have good data on that from the mouse studies that is actually what occurs. And we actually have a model for that in humans, which is impractical to go forward with. We think it occurs in humans also.
Malaria is complex not only individually, but it varies throughout the world
Right.
in the type of germ that's produced. Do you think that one DNA vaccine will work against all, or do we have to develop specifics for the different regions of the world?
That's a great question. And there's 2 problems with, now if we're getting to malaria, and it can be extrapolated to other diseases, but very specifically for malaria. One is that, as you point out, the targets, they're called epitopes, but they're very short peptides. The targets of these protective T cells vary from place to place, from parasite to parasite. In addition, the T cells responses that you make are actually different than the T cell responses that I make because our genetic background is different. So for me to be able to protect everybody in this room against 15 different types of parasites, I have to build redundancy into the vaccine. Thus, I have to make it complex. And I don't have a technology other than DNA to do that, and that's why I'm making it 5 genes, each of which has maybe 500 amino acids, and those targets are only 9 amino acids long. Nobody knows how much redundancy you need, but we certainly know you need it. Otherwise, the parasite will eventually change itself and evade your immune response.
How do you get the DNA?
No, the way that we get the DNA is this. we take the parasites and we can actually grow them in culture in the laboratory. We then extract all of the DNA from those parasites, and then using different technologies, but one of them is called polymerase chain reaction, or PCR, we actually amplify the gene of interest, the DNA of interest from that gamish. And we actually have it. I mean, you have it right there. You can't see it, but you can see it if you're a molecular biologist by tools that they have. And then you take that gene. So I was talking about the 5 genes that we're interested in. We took a whole gamish of Plasmodium falciparum, that's malaria parasite DNA, which we extracted the DNA, and then we amplified out those 5 different genes. So we have them, each one in a different test tube. Those different genes are then placed into the DNA plasmid, which is the backbone of the DNA vaccine. So if you envision a circle, and we actually had some posters here, but they didn't show up. We'll have them afterwards. But basically envision a circle, and it's open. And you take the gene you're interested in and put it in here, so you have a complete circle. And over here on the circle, you have some DNA which promotes the expression. Oh, here they are. Good question. Well, we're ready for it. Okay. Well, this doesn't really, it's the first I've seen it. It's a small circle, and in the circle there is the gene that you're interested in, and then in front of that is something called a DNA sequence. This is called a promoter, and it actually, we know about it, and it promotes the expression of the DNA in the cells of the individual. And at the end of that, there's another sequence which is called the terminator, and it stops the expression. So you only get the expression of the gene you're interested in, and the rest is a little bit of basically provides structure. So it's a circle. Oh, I'm sorry. So it's a circle of DNA.
So in the case of malaria, you physically take it out of the mosquito?
No, we actually grow the malaria parasites in culture. So that just like all of our cells have all of our DNA, all of the malaria parasite cells, no matter what stage they're at, have all the full complement of DNA.
You touched on the potential risks of DNA. You're actually taking DNA, you're putting it into our body, and then fooling our body into making foreign matter. Talk about what the risks are of that.
So let me just back up and say that when this technology was first talked about in the early 1990s, many scientists in the world basically dismissed it and said that it's impossible that you could take this fragile DNA, and it's called naked DNA because there's not mixed with anything. It's put in salt water, inject it into somebody's muscle, and that you would get the DNA actually to go in there without degrading and produce this protein. After so many scientists duplicated that work, it was clear that it was real. But I have to tell you that the first scientists to present this were practically laughed off the podium. Then the question came up, wow, you know, you're using recombinant DNA, you're injecting DNA into people, you will never be allowed to do this because it's inherently unsafe. And what was brought up was what is still a major concern is what happens if the DNA that you inject actually goes into the recipient's chromosome. So incorporates into into the host DNA. And 2 things could occur in that event. One is you could get what's called insertional mutagenesis, where actually the host's DNA gets changed, it gets mutated, and we know if that happens at a high rate that there's a good chance of cancer occurring. Okay? The other event that could occur, if the DNA got into a cell that circulated, say, to the ovaries or testes, is it possible that you could get that, it's called integration, integrating into the chromosome, you could get that occurring in your reproductive cells and it could be passed on to the next generation. So that there has been an enormous amount of work done in animal models to determine whether this occurs. And the answer is that we would never have been able to do the human studies if we hadn't proven. A lot of work's been done at Vical, at Merck, at different companies around the country showing that this doesn't occur. In fact, if you step back a second, and we talk about there's a field called gene therapy, where actually what you're trying to do is just that. So that the vehicles of the DNA in gene therapy are designed to try to get in and stay in the cell in most cases, and in some cases integrate into the chromosome, and it's been very difficult for that to occur. In the case of DNA vaccines, these plasmids here, these circles, are stripped of every signal, every bit of DNA that is known to be associated with that integration process. So they're actually designed not to integrate, and all of the scientific data thus far indicate that they don't integrate. Before a DNA vaccine is licensed, there will have to be, obviously, like any other new product, long-term animal studies demonstrating that that this doesn't occur when the animals are subjected to, enormous quantities of it. But right now, there's no evidence, and the Food and Drug Administration has been involved with us for almost the last 5 years in developing guidelines for testing this. So they were on board very early, involved with all of the procedures that needed to be done, and of course they don't allow us to go forward into humans without these studies having been done.
Back to the testing for a minute. The group of volunteers that are in your study, were they challenged with malaria?
They were not challenged
Okay.
with malaria.
So the group that will be in the study of the 5 gene and the 15 gene, it would be the same type of study you did with this initial group?
Right.
Okay, and then you go from there to challenging volunteers with malaria and taking it to the natural
Right.
exposure? Okay.
What is challenging volunteers with malaria?
Excuse me, yes?
What does challenging volunteers with malaria mean?
What it means is you take a ice cream carton with 5 infected mosquitoes and you put it on the volunteer's arm and you allow them to bite until they've actually shown about 5 minutes, and then you dissect them to make sure that the ones that actually are engorged, have blood in them, have malaria parasites in their salivary glands. And when the individual has been exposed to 5 of those, we have a 100% chance that individual will come down with malaria.
What type of timeline are we talking about?
So the timeline for our work, which is in the malaria area, is that we expect to begin the human trial of the 5-gene vaccine next summer. Probably the timeline now calls for August 1st, and we hope that we'll stay on that timeline. A lot has to do with our partners at Vical who are manufacturing it. For the 15-gene, we're about 6 months behind that. And then the study in the case of the 5-gene will take about 8 months before we have the results. And in the 15-gene, where actually the 15-gene vaccine, it's a bit complex to get into this. In contrast to all the vaccines that any of us have received, in which the vaccine that we receive is the same vaccine that children in the developing world receive, many malariologists believe that because malaria is so complex, there may need to be several different types of malaria vaccines. One vaccine would be vaccine for non-immunes, such as us, travelers, military personnel, people who are born in Nairobi and their children have never lived in a malarious area, but then they go home to visit the grandparents on the shores of Lake Victoria. And we're aiming the 5-gene vaccine at that group to protect children from dying in sub-Saharan Africa, where 2 to 3 million of them die every year of malaria. We think is going to require a more complex type of vaccine. And thus, to actually prove that that vaccine works, it will have to be tested amongst the population that needs it. That's a much longer-term prospect, and we're talking before we would get the results of the first really good field study would take 5 years probably. So it's a very difficult long-term process for which we need continuous enthusiasm and resources.
Captain, how complex a vaccine would you need for immunizing U.S. troops?
Well, we're hoping that the 5-gene will work, and it's targeted at the liver stage of the life cycle over here, which, can I talk and walk or not?
If you're going to do much explanation, you have to, and that
The
will
broadcast guys can't.
Okay, so I'm going to have Commander Carucci here point to the stages. In a virus or a bacteria, they don't really change once they're in your body, okay? And so if you make a good immune response against any part of a virus or bacteria, it works whenever that virus or bacteria is in your body. In the case of malaria, where the parasites are injected by mosquitoes, and then there's something called the sporozoite stage, which is in your bloodstream for about 5 minutes, outside of cells, before it enters liver cells, where it develops in a week to a mature liver stage, and the sporozoite stage has 1 nucleus. The liver stage has 10 to 40,000 nuclei. So there's this tremendous amplification process. It then ruptures out into the bloodstream, where each of those single nucleated, they're called merozoites, can invade an erythrocyte, and every 48 hours amplifies 20-fold. It's only at the blood stage that one gets the disease of malaria. So in the case of non-immune travelers, our goal, or military personnel, is to prevent the parasites from ever getting, it's pretty good, you were one step ahead of me. Is to prevent the parasites from ever getting out of the liver into the bloodstream, because there are no symptoms, pathology, signs of malaria associated with that. So we're focusing right now on the 5 gene there, but in a few weeks we're actually going to be having another publication in Science, I don't know if that's embargoed, on sequencing the genome of the malaria parasite, the first step towards that, and we believe that we will find many other targets at the liver stage and the blood stage. And again, we have no way, other than DNA vaccines, for capitalizing on these new findings from genomics, which is a whole nother subject. There is no technology in 1998, vaccine technology, which allows one to rapidly assess tens, hundreds, or thousands of new genes or proteins other than DNA vaccine technology. So it actually goes sort of in parallel, very complementary to this other field which is developing, which is the genomics field, where we are sequencing the genomes, all the genes of these different infectious agents, as well as sequencing the human genome. Yes.
Is your expectation that these vaccines will be something like permanent, or will they be time-dated like many of the current travelers immunizations are?
We would hope that we can build enough complexity into it that they'll be permanent. But since we don't have anything, and it's been 100 years since people have been trying to make malaria vaccines, it would be very premature of me to say that it would be permanent. I'd be happy if we have something that works for a year.
What is the risk that if you go from 1 gene to 5 to 15 and on up, that you increase the risk of giving these vaccines that you mentioned earlier, that you'll trigger some response unanticipated?
That can only be determined experimentally in people that you give it to. The data, any of the minimal data that are available thus far would suggest that that's not the case, because the risk of one of these events occurring when you give, say, 100 micrograms of 25 genes or 2,500 micrograms of one is likely to be pretty much the same. And it actually may be reduced.
I wanted to pick up on the money. What monies are needed? What monies are dedicated already?
Okay. If we talk about malaria now, you're talking about malaria money. Malaria is the most important parasitic disease in the world. If it's not the most important infectious disease in the world, it's amongst the top 3 with TB and HIV, tuberculosis and HIV. It kills 2 to 3 million people a year, and it infects 300 to 500 million per year, as estimated by the World Health Organization. The entire world's malaria research budget, that's everything, not just vaccines, was estimated by the Wellcome Trust 2 or 3 years ago to be about $80 million. If you contrast that to the amount of money the United States is spending on HIV research, which is in the billions, and you look at the magnitude of the problem, you also look at, from the military's point of view, the magnitude of the threat. In the 20th century, in every military campaign that the United States has been involved in, where malaria was transmitted, we've had more person days, more casualties lost to malaria than to hostile fire. That included World War II, that included Vietnam, and it includes smaller operations such as Operation Restore Hope in Somalia. The number one cause of casualties was malaria. So that's why malaria vaccine development is the number one ranked medical research, science, and technology objective for infectious diseases in the military. So that the amount of money that's being invested in solving this problem is minuscule in comparison to the magnitude of the problem and what is truly needed. And because it's malaria, it's very difficult at this point in the operation to get large-scale or any major pharmaceutical investment. So that we operate at a level, the DoD budget for malaria vaccine development in the military infectious disease research program is $5 million a year. For this, we were able, from the Office of Naval Research, to win what's called an Advanced Technology Demonstration Grant, which allowed us to go forward with this, which increased our budget by about 50% a year, but it's only a 3-year project, and we're in the second year right now of it. We were able to compete with Boeing and Grumman, and so on, and we have the only medical advanced technology demonstration in the Navy, but that's a time-limited operation. We only have it for the next 2 years, and again, that's allowing us to go forward. We were able to get $1 million Federal Defense Laboratory Diversification Grant several years ago with VICAL that allowed us, so we were kind of piecing it together, but at a far from efficient level.
And so what are you asking for? What would you like to see?
We'd like to see about $100 million invested in this a year. Yes?
If the 5-gene vaccine is successful, if the test on the human beings is successful, will that then automatically translate to use on troops? Will we start seeing that?
That would be, well, the process of going from experimental confirmation in a study, as I showed you, to licensure by the Food and Drug Administration is a very long and winding road, which requires multiple studies with many, many more volunteers to demonstrate safety and efficacy, so that we would then have to move to expanded studies both here and overseas prior to getting like a new drug application being submitted to the Food and Drug Administration for licensure. And as I think you're well aware from some of the experiences that we've had, no one is very interested in subjecting our troops to vaccines that aren't licensed by the Food and Drug Administration.
How long do you think before we might see it in use?
If everything worked perfectly, I couldn't imagine that one could get licensure in anything less than 5 to 7 years. All right, but that's saying that things work absolutely, and I'm not saying that they would.
You mentioned that in every place where we fought in the 20th century, where there was malaria present, that the casualties have been greater than battle casualties.
Right.
Isn't, though, the treatment of malaria such now that the risk is manageable? I mean, we don't have troops getting seriously ill with malaria, do we, anymore?
Well, there's always a few of them, and again, since Vietnam, we haven't really deployed in large scale to areas where malaria was transmitted. And I would add that we have never deployed to sub-Saharan Africa, where the intensity of transmission of malaria is 10 to 100 times greater than it was in Vietnam. Okay. When an individual, the problem to the military in terms of malaria is that in most cases we can say, in the vast majority, we will prevent the military member from dying. However, that person is knocked out of operation. And in both Vietnam and in World War II, we lost entire battalions. I mean, you have a certain percentage of the troops knocked out, and you render totally ineffective the fighting force. And that's why it's such an important problem. Yes.
Could you introduce
Yeah.
us real quickly?
Yeah, I'd like to introduce the members of the team. This is Dr. Rubing Wang, who is the first author on the paper that's going to be published in Science, and she is originally from China and now works full-time in the malaria program at Naval Medical Research Institute. This is Commander Richard Hedstrom, who in 1992 made the first malaria DNA vaccine with his own hands and has been an important, critical part of this effort since its inception in 1992. Commander Dan Carucci, who leads our malaria genome sequencing effort, which is going to be published in a few weeks, the first major advance there. And Dr. Alan Schreiber, who is the Chief Executive Officer of Vical Incorporated, a San Diego biotech company with whom we have been collaborating for 5 years on this project.
Doctor, one of these articles that I was reading in the packet says that in 1987, to test another vaccine, you allowed yourself to be exposed. Do you plan to be exposed in this test?
The answer to that is no, and the reason is that I'm actually protected against malaria. I mentioned briefly that there's a way of protecting against malaria, that if you take the malaria, these sporozoites, that was the stage that's injected by the mosquito, you take mosquitoes that are full of those and you irradiate them, which weakens the parasites. It allows them to go into your liver and then stop. That if you're exposed to 1,000 infected mosquitoes over about 3 to 4 months, you will be protected against malaria. And the protection lasts for about 9 months, which is what we're after for the travelers market. And we use that immunization process to try to understand the immunology and understand which genes are important in the protection. So a number of members of our laboratory, Commander Hedstrom and I, have been immunized with that irradiated sporozoite vaccine, and thus testing the other vaccine on us would be useless because, in fact, we're already protected.
On the video you handed out, was it Commander Hedstrom who's getting bitten with the mosquito? Yeah.
And was that the culture in that video, that red, I guess it was you were working with a...
That was a culture of lymphocytes, of the killer T cells. The culture of the parasites looks quite similar.